Students, welcome to lecture 11 in biopharmaceutics, and today's topic is protocol of bioavailability studies, and it is part 2. I'm your course instructor, Dr. Shabnam Nazir. So outline for today's lecture, it is part 2 of the previous topic which we discussed in the last lecture. And if you remember, in the last lecture we discussed about the protocol of bioavailability studies. So in this lecture, we basically going to continue the same topic, and we will complete in this lecture that topic. So what we are going to discuss in this topic is firstly, we will get to know about the study subjects and the inclusion criteria for these subjects, then clinical procedures or what you also call clinical trial, what are the various basic requirements and infrastructure for conducting these procedures, sampling or washout period, then test and reference products, as we know that test and reference products are very important whenever bioavailability studies are being conducted, especially the bioequivalence studies. There would be a test drug and one reference product with which the test drug is being compared for its bioavailability profiles. So the last one is the data analysis. These are the various steps which are the part of protocol of bioavailability studies. Subjects. Study subjects which are made part of bioavailability studies could be of two types: healthy volunteers or patient volunteers. Patients are selected as volunteers rather than the healthy volunteers. There are certain advantages associated with this choice. Inclusion of patients as volunteers shows rare conditions of usage of drugs. That's why the results are more authentic and reliable. Patients are getting free treatment and being get cured for the disease, for whatever disease they are being treated for. So this is additional incentive they are getting during the treatment process, which is actually the clinical trial. Results showing therapeutic efficiency of drugs are quite accurate because, as we just mentioned, that as study is being conducted in patients, so they are simulating or they are basically exhibiting very original or real states or disease states. Results are more authentic and reliable, and drugs therapeutic efficiency can be better tested during such conditions as compared to the healthy volunteers. Then inclusion of patients as volunteer avoids ethical issues of administering drugs to healthy subjects. There are certain drugs which are, when administered to the healthy volunteers, as they are associated with certain unwanted side effects, which we do not want to see or do not want the healthy subjects to experience them. So that's why the ethical issues arise along with that. For example, in case of antidepressants or antipsychotic medication. So for the trials of such kind of drugs, it is important that the volunteers or the subjects are patients than the healthy subjects. So how various disease states might affect the absorption patterns can be effectively evaluated. Healthy volunteers won't be mimicking such conditions of disease states. So, having patients as the volunteers is associated with this advantage that you are evaluating at the very same time simultaneously that whether how the bioavailability patterns could be affected because of such a specific disease state. Preferred for multi-dose availability studies. As we just discussed, that as it is being conducted in patients, and patients are the one who are getting multiple doses for any drug, not usually the healthy volunteers, and not they would be ready for that. So another advantage of choosing patients over the healthy volunteers. But there are certain disadvantages definitely are also. And first one is patient's arrangement in a certain number becomes difficult. There are certain diseases for which, if we are in the search of patients, it gets really difficult to arrange the patients in that sufficient number, which you also call the subject size, which is the required one, and the results will be more authentic with that number. The second problem associated with patients' inclusion as volunteers is that patients might be at various stages of diseases. Now, comparing the different patients who are at different stages of the diseases could interfere with the final results or their interpretation. Too many blood samples for longer hours from patients becomes an ethical issue, as we discussed in the previous slide, that along with the advantages, there are certain disadvantages also associated with the inclusion of patients is that um, whenever the too many blood samples are being taken, which becomes a need in case of certain drugs because of their longer elimination half-lives. So in that situation, now this would be an ethical issue that as the patients are suffering already, so whether taking too many blood samples too frequently is right or not. Then unethical to administer dosage form to a patient whose therapeutic efficacy is not known. Sorry. Another ethical issue that the drug which is being administered to the patient, we certainly don't know sometimes if especially the formulation is new, it is a new dosage form, or it is a new drug. You don't know about its possible effects which it could be having on the body. So that's why most of the time certain drugs are tested in terminal patients, which might possibly be associated with unwanted side effects. Um, Bioavailability of the test drug will be affected by the drugs already used by the patients. As we just discussed, that these are the patients which are being included as volunteers. They definitely using other drugs because they are being treated for certain disease condition. So in such a situation, they might be administered with other drugs. So those drugs might interfere with the one which is under the test. Procedure. In order to understand the clinical procedure, it is important to understand study conditions, and all volunteers should be on similar diet. It is very important that whoever volunteers are part of the study, they are getting the same diet, because otherwise different diets, ingredients of different diets might interfere with the test drug and so the final results. No administration of any other drug 1 week before the study, and purpose is the same, that no interference of any other drug with the test one. Nature of diet allowed, amount and volume of fluid intake allowed is a set patterns which is decided prior to conducting the study, and during the study one should stick strictly to the standard pattern approved. Test and reference products. Both the products are tested for in vitro dissolution profiles, and their in vitro dissolution profiles should not be significantly different. If there is a significant difference between the dissolution profiles of both of the products, heading towards a clinical trial is absolutely out of question, because both of the products, failed even the initial test. Period. What is a washout period? This is a duration or a time period which is involved between two treatments, two clinical trials, or two segments of the study. And it is also called the time interval between two treatments. And what is the a very specific criteria for determining this washout period is that it should be equivalent almost to almost 10 half-lives between two treatments. Let's suppose a drug has a half-life of 2 hours, so it means that there should be at least 20 hours involved in the next treatment before you conduct the next clinical trial. And Another very specific characteristic property of the washout period is that it should ensure 99.9% elimination. So means the maximum carryover effect that it is allowing is 0.1%. And it is a function of elimination half-life and dose of drug administered, and at least it should be 1 week. Then comes the sampling. Sampling is the appropriate frequency for sampling can be decided in a way that it should cover certain specific parameters. For example, the number one is the Cmax. It must cover the Cmax, which is also called the maximum concentration. And secondly, it should be able to cover the Ka, which is absorption rate constant, and area under the curve. Area under the curve we know is a very important parameter for the assessment of bioavailability. So the sampling frequency or the data point should be such that it should be covering the Cmax, Ka, and area under the curve. Then elimination phase should must be covered also uh, by with the help of these sampling frequencies, and so the important parameters which we are mainly concerned with is the Cmax, absorption rate constant, area under the curve, and the elimination phase of the drug. So data analysis is associated with certain objectives. First of all, it helps us to find out the significance of difference between the bioavailability profiles of test and reference products. Basically, bioavailability study is a comparative study in which we are comparing two products for their bioavailability profiles. So if there is a difference that must be identified. So let's suppose a difference has been found. Data analysis also helps to understand the extent or the nature of the difference between the values with the help of various statistical parameters like standard deviation or the variance. The standard deviation help if, for example, the standard deviation value which is associated with the mean value of your results, if high, so it is showing that there is a great deviation between the values of your data points or the data set, and the values are highly deviant from each other. So standard deviation in this way is showing there is a problem with the data set. And to understand the source of differences, like whether these differences which we are finding with the help of data analysis between the products, what could be the possible source, problem source for such differences, those could be assessed with the help of a suitable analytical, statistical, or data analysis technique. So different statistical methods are used to find out the source of variation, and with the help of data analysis or using a suitable statistical approach, the identified variables effects are also evaluated, like how the different variables which have been set, and the studies basically being conducted to observe the changes in the variables, whether the changes in the variables have any impact on the study parameters or not. So data analysis is also helpful in that connection, and it also helps to calculate the required parameters. And most commonly used tests which are used for the data analysis according to the nature of the study, statistical one, are t-test of significance, t-square test of significance, and analysis of variance test.